Thank you, ma'am. It gives me immense pleasure to introduce you all to our guest, Mr. Bhausaheb Patil sir. Mr. Bhausaheb Patil sir is presently working as a head business operations, decentralized clinical trial solutions at IQVIA. Sir has completed his middle management development program, general management, from one of the prestigious institute, Indian Institute of Management, Ahmedabad. Sir has completed his post-graduation in pharmaceutical sciences from Bedla Institute of Technology. Sir has completed his bachelor's degree in the field of pharmaceutical sciences from Shivaji University. Sir has worked as head clinical operations, quality management Asia for about 2 years, also as director clinical operations at Singapore, Indonesia, and Vietnam for a year. He is also the head of CTL Services Southeast Asia and Korea and Director Clinical Operations Indonesia for about 1 year, 3 months. Within 25 years of his work experience on various roles in IQVIA helped him to get deeper insight of productivity, delivery, and quality, which are the key drivers associated with clinical research industry. Dr. Bhausaheb Patil sir has have worked in various variety of functions such as clinical monitoring, clinical operations, and project management, financial management, resource management, quality management, and risk-based monitoring approach using concepts of artificial intelligence, machine deep learning techniques, and also got exposed to direct line management area and interaction with senior management teams within IQVIA. With this brief introduction, on behalf of Bharati Vidyapit College of Pharmacy, Kolhapur, we once again welcome you, sir. Now we kindly request Mr. Bhausaheb Patil sir to proceed with his session on pharmaceutical quality system for product life cycle management.
Thank you. And I'm just going to share my screen.
Yes, sir.
Please give me a second. And
Yeah, sure, sir.
yeah. So can someone please confirm if you can see my screen?
Yes, sir.
And I'm just recovering from a normal cold, so my voice is not as usual.
It's okay, sir. But you
Hopefully
are
it's
clearly audible and there is no issue, sir.
very good. So first of all, good morning, everyone. It's early morning for me, about 5:30 a.m. in the UK, but very happy to be here. So first of all, thank you so much, Dr. Manish Bhatia, Mrs. Dr. Bhatia, and all the organizers of this program. I think this is my second sort of presentation to similar program. The
Yes,
first one I conducted back probably exactly a year ago. And
absolutely, sir.
that was more into the use of artificial intelligence, machine learning into the clinical trial. And that's the exactly work that I have been sort of overseeing right now in my new role, which started literally 4 weeks ago. Uh, Heading the business operation for IQVIA in the decentralized clinical trial world. And probably those who were part of my uh, talk in the last year, you may know usually I would like to keep this presentation more engaged. And I know we are challenged with this virtual world, but hopefully either you can provide some of your responses in the chat function, or if there is no background noise, at least provide your inputs. So what I'm going to do, I have a few slides and the topic which is given to me today, pharmaceutical quality system for the product life cycle management. And Dr. Bhatia has challenged me to complete this topic in 2 hours, which is great. Typically, I think there are different components of the quality system. And if I had to choose one component and just go even superficially, it would take probably 3-4 hours. And there are many such components, and I'm trying to finish that in 2 hours. Hopefully, I will do the justice. But if if you have any questions, we will see if you want to ask, use the chat function. And hopefully, I think Dr. Bhatia, we will have someone to read out those questions, and I will do my best if I can answer them.
Yes, sir.
Okay, so so thanks again. And before we go start, belated Happy Pharmacist Day to everyone.
Yeah, thank
And
you so much, sir.
anyone knows why we do celebrate World Pharmacist Day? And I'm just asking the question, and if somebody, if you want to just use your chat function just to give your thoughts as to why do we celebrate World Pharmacist Day? And if somebody can read out if there are responses, if there are no responses, that's absolutely fine. And while I think you're making your responses, actually, I'm currently working from home. So that's my little office, what you see on the first floor on the left side. That's the place I'm talking right now. This is not the typical scene, but I think about 4 years ago, we had I think one of the biggest snowfall in London, and that time I just took the picture. It's very rare to see. So, and that's my family, my wife, my 11-year-old son, and the another son who is playing with the snow on the right-hand side. He's now into the university. So any responses we are getting, Dr. Bhatia? Why do we celebrate World Farm?
No,
Okay.
not until now, sir.
Okay, and absolutely fine. And the reason I asked that question, I think that's the curiosity. Uh, I think I usually have built in, and I'm sure all of you have. So before we do anything, we need to know why. And that's exactly sort of coming back to quality system. But uh, just for your information, there was an International Pharmaceutical Federation Council, and which was established in Turkey in the year 1912. So almost about, I think, close to more than 100 years ago, 110. And they actually, it was established in 1912 on this day, which the 25th of September. And in 2009, they had the World Congress of Pharmacy and Pharmaceutical Sciences in Istanbul, in Turkey. And they decided to use this day to celebrate, to promote the role and actions of pharmacists in promoting health across the globe. And in that memory, we celebrate World Pharmacist Day. Uh, Now, just taking that ahead, I also, maybe want to take it ahead and understand the pharmaceutical quality system for product life cycle management. So there are two terms we are using. One is the pharmaceutical quality system, and another is the product cycle, product life cycle. So before I think it'll go ahead, I just want to quickly, what I'm going to contribute based on my limited experience of the last 25 years, and I'm still learning. Uh, I have sort of managed variety of roles within the clinical research industry, and quite happy to say, fortunate enough that I made the entry into clinical research when the clinical research was just starting in India in the year 95, 96. And I think that gave me multiple opportunities, not only just grow within India, but then move to Singapore, contribute my learning at the same time, learn the way clinical research is managed across Asia Pacific. And I think some of the world of clinical research is quite fascinating. It is changing very fast and changing for good. And that has also resulted in terms of thinking about, I think there were a lot of terms used by Dr. Bhatia: innovation, the quality system, and that also it gave me a lot of exposure in terms of be part of building some of the quality system, and also be part of how we can speed the clinical research by bringing some technologies. So you may have heard of this risk-based quality management or risk-based management. What is that? So I think this is something, rather than being too theoretical, I got an opportunity to operationalize that. And I think I consider myself quite fortunate. And I think it is quite interesting sometime to say, the way you operate in India, the way you operate in Singapore, the way you operate in the UK, and the way you operate in the US. It is just not the location, but I think there are a lot of things come to mind. And I think Dr. Bhatia also spoke about what is the quality system. It is not just about system per se, because ultimately at the end of that system, somebody needs to build that system and somebody needs to operate that. And then that system is going to give you that output. So there is also a lot of people involvement. And so I'm going to contribute some of my experiences. So this is just a slide just to let you know what keeps me busy and awake at night. Uh, and of course, I think, maybe if I start with some of that first part, yes, I think, that keeps my family and my upbringing. I was just making a joke. My elder son, he started his university last year, and for the first time, he started living outside of my house. So as a parent, you are always worried because we know what we have done probably when we were child, started to live in the hostel. But yeah, I think that's my family and their upbringing. I think that's what keeps me awake. I also usually like to run for better thoughts. When I say now, I don't run, but I usually walk because that gives me different ideas of innovation, how same work can be done differently. I do watch cricket. That's again to rejuvenate myself because you need something. You cannot just work all the time. Uh, and yeah, I think probably this is something I usually would like to have my workplace clean and even my house. So typically on some of those weekend, you will see with my shorts and cleaning my house, the toilets, and that's typically what we do here. You end up doing everything right from washing the car, cleaning your house, cleaning the garden. But I think that's has helped me to make self-dependent, and I think as I do all of that, yeah, that is sort of giving me that energy to look at some of the quality and compliance metrics, the customer complaints, doing some of this innovative part, be part of those investigations, understand some of the data behind, also understand how the clinical research was done, how clinical research is done today, and how it can be done differently. And I think that's where probably, if time permits, I want to talk about what I've come up with my own model. I call it as an improve, and that is very closely linked with what we are having the discussion today on the continuous improvement problem solving process in a just to improve quality. So let's talk about those two concepts now, the pharmaceutical product life cycle. And if I look at the pharmaceutical product life cycle journey, it it really based on, in my opinion, three pillars. So any product life cycle, and be it a medicine, any type of medicine or medical device, it's the tablet, capsule, or an injection. Uh, Whole development is dependent on three areas. One is it's people who are involved in that development. It is also the process which is followed by the people, and the technology used along the journey of that product life cycle. And uh, all the discussion revolves, so many times people say people, process, technology. Some organizations say people, process, tools, or people, process, systems. And I think those are used into this pharmaceutical product journey, which really starts, if you look at in the lab, and I'm sure most of you as a researcher must have been involved into that basic research, and that once that basic research gets complete, your product is born, and then that product goes through series of development right from the time the preclinical studies or the laboratory studies, during which the aim is really try to establish the dosing or toxicity level, going through then phase 1 study to understand even before finding if the product is effective, if the product is safe, and gather that safety information, then moving towards the phase 2 once the safety is established. understanding if it is effective, at what dose it is effective, going through, in terms of then phase 3 study, which is safety and efficacy. So looking at both, but across multiple ethnic origins, different age groups, different sort of types of subjects of clinical trial participants. And once all that safety efficacy is established across various parameters, that is the time when drug gets approved by FDA or by the regulatory agencies like in UK, the MHRA or the European Medicine Agency across Europe, the PDMA, which is the Japanese regulatory agency, or in Indian context, it is the DCGI. And I think that is the time drug is sort of ready to be launched into the market. And then you have that drug is consumed by various individuals, or the normal the population who needs that. Again, the journey doesn't stop there. You also have the phase 4 trials just to look at the post-marketing surveillance, whatever are studied during the phase 1 all the way to phase 3. Are we seeing any other impact of the drug during the long-term use? Are there any additional side effects? At the same time, can we use this drug for any additional indications? Or can you use that drug for the same indication, but in a different age group or different type of population? And now, if you look at, one of the most recent example, the vaccine development, it has gone through all these phases. And again, that vaccine, so we have some of the people who developed that vaccine, they followed the processes. And one of the significant change which happened last year in the vaccine development, that was more on using the technology in order to speed the clinical research, at the same time not compromising safety, but really focusing on that in addition to safety aspect, how we can speed that research and make sure we have these vaccines available. So again, I believe as a pharmacist, we should be really proud of what we have done as a community across the globe. And I would like to think we are the reason why the world is living today the way they are living today compared to last year. Almost like, if you remember when the pandemic hit, all of us were locked down without knowing what's going to happen tomorrow. The future was quite blink. And I think that's where, as a pharmacist, we were given that a directive as a civic responsibility, do something about it. We need some effective treatment to eradicate this invisible killer. And I think that is what most of this pharmaceutical company, the pharmaceutical community across the globe, they did it, bringing these various vaccines into the world market. And I think you are saying that is helping us. I'm not saying the COVID pandemic is over. It is still ongoing. Maybe it is changing from pandemic to the endemic phase, but the things are quite different. We know how this virus behaves. We know what needs to be done to get rid of this virus. And we know what this virus is going to come tomorrow. And I think that has better prepared us as a pharmacist to see how we can help world to live safer. So I think hopefully you have this idea now, the pharmaceutical product journey, and I think it is quite complex. Again, as Dr. Bhatia mentioned, it requires a tough sort of, it is highly regulated industry. And I just want to pause here, and the quality is of paramount importance. So how do you define the quality systems, or maybe quality for a pharmaceutical product? Any thoughts, if you would like to just use your chat function on Teams and just say, in your opinion, how would you define quality in a pharmaceutical product? And I have some of my own thoughts, but that's where we will move towards. So maybe, as if Dr. Bhatia, somebody, if you can please read it out, because I cannot read both screens at the same time. If you can just take some time and define in your own ways, what do you mean by quality? And I'm just going to take a pause, and Dr. Bhatia, making sure my voice is still audible and you can hear me okay.
Yes, you are audible.
Audible, sir.
Very good. Any thoughts? How do you define quality? Okay. If
Yes,
maybe
sir.
like I will.
Fonts are visible, sir. No problem.
Okay, good. So I will move ahead. Now, the way I define quality in pharmaceutical product, I think, and again, this is not a textbook definition, but my definition, something I would like to think. Uh, I think you can measure quality in other service line, maybe like how, and I'm just using probably an example. What was the quality of cell? And maybe like and the cell maybe in the pharmaceutical industry or the cell in a car manufacturing industry. And in terms of you measure that cell in certain finite number. And that number could be like, yes, my cell was 80% good quality, 100% good quality, or maybe 120%. And you measure sort of, in some absolute number. However, I don't think you can measure quality in a pharmaceutical product that way. And the way product needs to be perfect. And because, once that product is consumed by the patient and for some of those life-saving treatment, you can only measure the success of that product if the patient gets better. So and again, in a clinical trial setup, I further elaborate quality as, either your patient is dead or alive. There is nothing in between. So success is not like, you cannot say patient is 80% alive, or patient is 100%, 120% alive. It is just binary, yes or no. And I think that's how I would like to compare the quality. It needs to be that much robust. It should be sort of perfect at its best. And therefore, the way as I see quality is not just a stagnant in pharmaceutical product journey. It is constantly evolving, and it is constantly raising its bar than what it is. And as I said, it is sort of highly regulated. And because it is regulated from the regulatory agencies, it is regulated by the people like we, and it is regulated by the end users who are consuming this medicine. So this is sort of the definition about the pharmaceutical quality system. We looked at the product life cycle, and I just used this textbook definition of pharmaceutical quality system. And again, I was glad Dr. Bhatia said, it is not about just process. It is not just about system. It is a management system. So in any given company, it is the management who drives that quality to direct and control the pharmaceutical company in terms of quality, with the primary objective to ensure that the final product, which is coming at the end of that system, it is of good quality, it is efficacious, and it is safe to the patients who are going to consume that. And this is where I think it's a journey which really starts from the management, from the top, and it flows bottom down in order to measure the performance of the business. And this is also not quality is not built in a later phase of the product life cycle. It is determined at the beginning. It is established at the beginning, and then it is it is sort of managed throughout that life cycle of the pharmaceutical product. So now what I want to do is I just want to talk to some of those pillars. And the quality is, I think it's a strong word, but what do you mean that quality by quality into the drug development process, right from the time it gets into the lab research, all the time it gets out. So there are different aspects. So this is just another illustration. So if you look at, you have basic research, which is like, I would say, not 100% regulated, because then you just try and test based on various development in the lab to understand the molecular behavior. And then through that you have the drug discovery, and then the drug goes to that non-clinical development, which is what the toxicological studies, animal studies, followed through the clinical trial, and then all the way to the manufacturing. And as I said, you have now we have some of the toughest quality standards in each phase of that drug development. So if you look at that non-clinical development, you will hear the term GLP, and that means the good laboratory practices. And I think I want to touch on some of these aspects during my later part of the presentation. What are the quality standards for that good laboratory practices? Moving to, when that drug moves from the non-clinical development to the clinical development, there is also another term, the gold standard, which is the good clinical practices, GCP. Once the drug passes through that clinical trial phases, the story doesn't end there. How that is going to be manufactured, and that is called as GMP, good manufacturing practices. And again, the story doesn't end there. After drug is manufactured, the drug needs to be dispatched to the various places, and there is
Dear participants, all are requested to put your mic off.
So thank you. So when the drug is manufactured, the story doesn't end there. And again, if you take example of vaccine development last year, these vaccines are temperature sensitive. So therefore, when the vaccine was manufactured, there was a quite Herculean, difficult task for the pharmaceutical companies, how they are going to dispatch that by maintaining that cold chain. So, and then make sure by the time vaccine reaches to the end user, it is still effective. And that is called as a good distribution practices or good. And then there is also another aspect, good documentation practices, which cuts across all the good laboratory and good clinical and good manufacturing practices. And sorry, I think what we're going to do, these are the quality standards. So, you have good laboratory practices, you have good clinical practices, you have good manufacturing practices, you have good distribution practices, you have good documentation practices. And these are some of those, what I would say, gold standards to maintain the quality system in the pharmaceutical product life cycle. And again, at the bottom of the slide, you will also see the distribution, how they, sort of go. And all this put together, the industry called this as an Gxp assessment. And when you say Gxp, it involves the GMP in terms of the manufacturing control, which is the good manufacturing practices. It also means GLP, which is the laboratory quality standards, the clinical research quality standard, which is GCP, and then the good documentation or good distribution practices. Because again, another important part for the vaccine development, you have, I think we experience, in addition to maintaining that cold chain, it is also important that vaccine gets delivered where it is needed. So it's the supply chain management. And I think these are all sort of quality standards. We will talk about each of these quality standards in the next few slides. So I think, and again, I'm not going through each one of these details because I think some of this you are familiar, but probably I will give you some of the operational aspects. So again, I'm not a GMP expert. I had sort of limited experience working in a pharmaceutical company on the manufacturing side. But if you look at that GMP, there are certain key areas that needs to be followed. So of course, at the core of it, you need to have a clear, defined processes which needs to be controlled all along. So, if you think about manufacturing a tablet, manufacturing in a capsule, right from the time how we are going to source the raw material, what is the quality of raw material, how frequently machines are cleaned or validated, how frequently the quality control is taken place, where it gets documented, how the people who follow that machines or use those machines are trained appropriately, where their training gets documented. Do you have process to manage any deviation? What does that mean in terms of if you're manufacturing tablets, then in that tablet manufacturing, what is the alarms that you are giving in terms of deviating? So for example, if the weight of tablet should be say 250 milligram, what is the deviation you are allowing? Is it like plus or minus 2 milligram, or is it plus, and then how you're controlling that? What steps you are going to take that? And then what systems you are going to follow that in case the deviation becomes unmanageable. At the same time, what is the traceability of manufacturing and distribution? If you think things are not going in the right way, how you are going to recall some, what is your recall procedures? At the same time, how you are going to conduct that investigation to see why things went wrong despite proper planning, and understand, conduct those root cause analysis to make sure you take what is called as corrective and preventative action plan. And this is like kappa management. And that is part of the risk management across various parameters. Again, most important criteria, clean and hygienic manufacturing area. How frequently that area is getting cleaned, especially in case of if you have vaccine development, or in it is not just the cleaning, but making sure some of the hazardous material is destroyed appropriately. And this is all part of control as a part of good manufacturing practices. If I look at this quality standard good manufacturing practices, the US FDA, they have defined this approach and they call it as a CGMP. So it's really, if you look at the history of good manufacturing practices, it started from, I think, almost in the year 1920 at the time of penicillin discovery. And it is still followed. You have heard some of those anti-cholesterol medicines developed in the last few years, anti-HIV treatment, and as recent as like last year in terms of the COVID-19 vaccines. So it is all followed, and they call it as a current good manufacturing practices. And there are sort of 7 critical concepts. These are followed into these good manufacturing practices. The number one, of course, the quality. Sorry, it's my wake up call, so I'm just going to ignore that. So it is called as the quality, which is refers to the strength, purity, then quality by design or product development. And this is also a new concept you will hear across the industry. And it is not just used for GMP, it is used for GCP, it is used for good documentation practices. And, so whenever you develop any product, how you design that product with quality mindset in the mind at the beginning itself. So proactively, you're thinking about what might go wrong. And therefore you build your product in that way. And just to translate, vaccines are time sensitive. So how you are going to control the temperature? Can you come up with a vaccine which is still going to be stable at the maybe like at the temperature which is not going to be too difficult? You take example of Pfizer BioNTech vaccine, which requires storage at -70. Now that may be relatively easy in the US or UK, where normal temperature is about 20 degrees plus or -5, and that is extreme case. But it is going to be extremely difficult if that vaccine needs to be used in some of the tropical countries like South Africa, maybe India, Singapore, because the temperature is quite high, and especially in summer, it is even higher. So how you are going to control? Yes, you can have cold chain, but at the same time, can you design the product which is going to be stable at those temperature? And that is just one of the example of quality by design. Then quality risk management. This is another concept which is sort of used in the GMP. How you are going to manage the risk. And there is a difference between risk and issue. So if you think about, what are the things which are likely to go wrong in my product life cycle, be it in terms of storage, be it in terms of manufacturing, be in terms of people training. So you think about those risks, anticipate them, and put appropriate risk management plan in place to ensure you avoid that risk to the extent possible. And if you cannot avoid the risk, if that risk becomes an issue, you have appropriate root cause analysis concepts in place, you learn from it, and you make your systems a lot better so that in future, same risk doesn't become an issue. And that is through sort of those corrective action and preventative action plan principles, change control. What are the things you may want to change in that in the process of that manufacturing? What kind of and it also, I think the US FDA guideline talks about 6 systems of inspection model, and those 6 systems are the quality, the production, the facility and equipment, the controls which you are putting on in the lab, as well as the material, what kind of systems you have in sourcing right material and making sure product is packaged as well as labeled appropriately. So again, I spoke a lot, but I think these are some of the regulatory guidance available, how you ensure you're following those current good manufacturing practices or principles. Again, talking more, and when Dr. Bhatia sent me the brochure of this this FDA program, I also looked at some of the terms I think that was referred, and I was really happy. What are these Q10 principles? So again, I will not go into detail, but if you look at the ICH Q10 guideline, these are sort of International Conference on Harmonization. So across different regulations that I talked about, they have formulated these common guidelines, what is called as ICH Q10 Quality Principles 10, which needs to be followed by the pharmaceutical companies as a part of their pharmaceutical quality system. And that is right from the time products gets developed all the way up to product gets produced and distributed. And if you look at the genesis of this ICH Q10, the quality system, it originates in the International Standard Organization, the ISO concepts. That's another concept which I'm not going to touch on that, but I believe you have additional speakers who will talk about that, the ISO 9000 certification. But some of these concepts are pretty much similar in what you see in ICH Q10, as well as there is another guideline which is called as ICH Q8, which is based on the pharmaceutical development. And there is another ICH Q9 guideline which talks about quality risk management. So you can see how complex the world is. We talk about quality system, and from that quality system, we said, we started talking about GXP, and within that GXP, we started talking about good manufacturing practices as one parameter. And within that good manufacturing practices, you have some of these guidelines which are defined in ICH Q10, ICH Q8, ICH Q9 in terms of quality risk management, quality systems. And these guidelines, again, just to put it in another way, these guidelines are around I'm just going to blow up my screen a bit, just going to use my laser pointer. So these guidelines are, about how the individuals are trained, how the documents are managed, are my systems validated? And when I say validated, even if as simple as if I'm taking weight of the tablet and it says 250 milligram, is that weighing machine validated, calibrated appropriately so that I can be assured that it is the correct weight that the system is giving to me. The change management control, and I think that it is also a lot about if you are changing any process in the system, how you are managing, documenting that change, how you are maintaining all the equipments. We talked about that. How you are providing oversight on the suppliers who are providing you the raw material, or who are providing you the machines. And then in terms of how you are self-assessing quality. And I think we will also talk about how that quality control is different to quality assurance. So this is sort of, it is all defined in industry. They call it as a quality by design concept, how you define at the front end. And it is taken from this ICH Q10 pharmaceutical quality system. So I think, we talked one portion about GXP quality principle, which is good manufacturing practices. And I'm just looking at the watch, Dr. Bhatia, making sure we are on track. And I believe we have another 40 minutes or so for my talk.
Yes, sir. Yes, sir. Sure.
Okay. Any questions so far? So before I move ahead and take this another second dimension of quality standard. So we talked about GMP. We will switch over to the GLP, good laboratory practices.
Sir, at least here on the chat box, because of technical error, participants are unable to share their question, but they are allowed to share their question on the WhatsApp. So as the questions we will receive, we'll share with you also, sir.
Thank you. So what I'll do now from the GMP, let's talk about the another quality standard, which is the GLP, and that is the good laboratory practices. Now, what does that good laboratory practices mean on the ground, how it is operationalized, and again, the how it is different from GMP. So good manufacturing practices is all about, when you manufacture a pharmaceutical product, and good laboratory practices, it is, I think, more relevant in the laboratory, say like, and that laboratory could be for the animal studies, which is done before the drug comes to the manufacturing process, as well as when you analyze any samples into the lab, be it conducting the analysis of a tablet or a machine, the capsule, and it talks about the some of the elements of good laboratory practices, like do I have certification of lab facilities, and there are also different types of certification. uh, uh, there is a American Society of Laboratory. There is also like an NLB, National Laboratory Practices certification, and those certification needs to be reviewed. And again, if you look at those certification, talks about what kind of processes each laboratory is following, how they can give assurance that if I say measure hemoglobin in the lab and it is giving me value of 14.9, is there a standard, and it gives me that if I test that blood sample maybe next time in the same lab, can it give me the same result? So or sometime nowadays in the clinical research setup, you have laboratories in the different parts of the world. So you have lab in India, you have lab in South Africa, you have lab in the US, and are these labs sort of following the same standard? And what does that mean if I test blood sample in one lab, same blood sample in all those three labs in the different part of the globe, can it give me the same result? And I think that's the level of accuracy that we are looking at, and putting those manufacture the controls, making sure you have appropriate statistical procedures in place. The Even how this things are getting documented, or how the specimens or samples are tracked, what kind of quality controls you are putting in place. And again, I think the documentation, it will come again and again in all those GXP concept. And the way I think I have understood in all my last 25 years of career, there is a saying in the industry, especially in the pharmaceutical industry, if it is documented, it is done. If it is not documented, it is considered as not done, even though it is done. So I think there is a lot of emphasis on documentation, and I think that is important, because that is only way you can show whatever you have manufactured or whatever you have done in the past. There is clear trail of event you can build in by reviewing those documentation. And that's is part of the quality system, to put right controls in place in the research laboratory and the organization. Now let's switch over to the GCP standard. And again, if you look at the standards, you talked about GMP, you talked about GLP. The standards, almost I think they talk the same thing, but slightly in a different way. So if you look at GCP, they follow this ICH E6 guideline, International Conference on Harmonization, Good Clinical Practice, Part E6. And they talk a lot about quality risk management. And it is again built on ICH's quality risk management principles defined in Q9 guideline. And I'm just trying to explain, probably like, this is applicable to, I think, most part of it. So in a quality management system, which follows really from the top, what kind of processes you put in place, and again, how you identify the critical processes. Now, what does that mean for a manufacturing? For the manufacturing, say again, I'm giving an example of vaccine. What is the critical process of vaccine development? Maybe temperature control, making sure that your vaccine is effective against variety of strains. So that's your critical process. Maybe in a clinical trial setup, if you have to design the protocol for a diabetes study, what is your critical process for diabetes study? Measuring the blood glucose value. So I think it starts from identifying those critical processes in any sort of practices. And once you identify that critical process, how you are going to identify that? So again, giving an example of vaccine development, If you think about your vaccine development, critical process is the storage, how you are going to identify your storage is appropriate or not appropriate. So that's part of your risk identification. Then going to, once you identify, yes, my product is time sensitive, how you are going to evaluate what is the tolerance level, if your product is stable. So is it like -70 plus or -2 degree, plus or -5 degree, and what does that mean? If my product suddenly jumps from -70 to 50 degree for 1 minute, is that still stable? Or I'm just talking about, even though that product is out, remains at -68 or -72, even for 2 minutes, its efficacy is lost. So sort of it is not just important to identify the process or identify risk within that process, but making sure you are evaluating with the right controls. And once you do that, how you are going to control that. So again, giving an example of this vaccine development, if you know that your vaccine is highly temperature sensitive, -70, you cannot allow to go even for a couple of minutes beyond outside of plus or -2 degree. How you are going to control that in terms of and again, think about this vaccine gets manufactured maybe in the US or in Belgium, and it's shipped all the way across to places like India or Singapore. So during that transit, how that cold chain is going to be maintained. So that's part of your risk control. And then how you're going to communicate about that risk control. So, in terms of, again, giving an example of this vaccine development, when these products are shipped out, you almost have this temperature logger. So even though you have this product in the on the plane traveling from Belgium all the way to Singapore, during that course of 10 hours, you have the temperature logger, which is constantly communicating through satellite communication and telling the end user as well as the person who are shipping the drug, saying that your vaccine is still within that desired temperature. And then there is somebody replacing that dry ice or mechanism to make sure nothing goes outside of the set boundaries. So that's second part of the communication. And then you have that review. In case that risk becomes an issue, then you make sure there is a proper process for product recall. And again, it is part of the good clinical practice. I gave example of vaccine, but how the protocol is designed in the clinical research, how you identify the right processes that you want to measure, how you evaluate them, and at the same time, if things do not go correct, can you amend the protocol? And that is sort of part of that the change management control. And this is where you have different type of controls which are put in place in some organization. Like, as I said, the quality starts really from the top, having that structured management review, how the management is going to review the quality. And then, in order to give them visibility, how they are going to look at some of those processes. So again, as an example, how they get an information that all the manufacturing tablet manufacturing machines are calibrated at least once in a week. Is there any machine which is not calibrated? Is there any machine which is showing sign of wear and tear? So how you get that almost like 35,000 helicopter view to make sure you are almost like producing the same product consistently, and you are almost in true sense you are inspection ready, and you are evaluating that with some of those what industry call as a key quality or key performance indicator. And again, you are constantly learning from your past and making sure you are making your quality system even better next time. You are almost like raising your bar. Exactly same is true in the good clinical practices. When you design any protocol, when you conduct any clinical trial, again, I think, as I said, clinical trial, it's an experiment, and therefore you have to be so methodical and plan well in advance. Think about the worst, and as I said, the success in clinical trial or in any part, you can't measure 80%, 120%. Either the clinical trial is failure 0% or it is a success 100%. There is nothing in between. And I think that is where you have some of these concepts of good clinical practice, wherein we talked about that and how you are going to identify critical processes all the way up to how you are going to report all those risks and learn from those. And this is just one area, there is a difference between quality assurance and quality control. So the way you look at quality control is more about detection. So if the things are going wrong, then you detect that through quality control. Whereas quality assurance is you think about how you're going to prevent that happening. And therefore, if you look at, the quality control is more focused on the product versus quality assurance is more focused on the process. So again, if you look at quality control is sort of looked at the end of the product life cycle, whereas the quality assurance is built throughout the process to make sure you will have less to react. You have less to manage, and therefore, the way quality assurance is considered, it is more of proactive. Quality control is more of a reactive. Again, quality assurance is more of a staff function. It helps you to prevent the defects, versus quality control sort of, it will help you to find the defects, but it will not help you to prevent the defects. So that's sort of another way to look at how these quality standards are managed. Now, I think we talked about three sort of quality standard concepts: the GMP, good manufacturing practices, the GLP, good laboratory practices, GCP, good clinical practices. Let's talk about the other two parameters, the good documentation practices. And again, as I said, this is an area you may have heard, the US FDA, they conducted some inspections of the GMP facilities or bioequivalence, bioavailability. They conducted inspection of the laboratories, and they issued some warning letters, or they decided not to use some of the end product coming out of those facilities for approval in other countries. And I think this is where what I have observed, there are many organizations, they follow most of these principles, the good manufacturing, good laboratory, good clinical practices. But I think most of the area where I think there is tremendous scope is the documentation. And again, as I said, the documentation, it's like, documentation before you decide to do any activity, how you manage that, during part, how you document with the change control, and towards the end, how you ensure that whatever was documented in the beginning, you followed that. And if it was not followed, what are the things that you have learned so that you can implement those learning in the next life cycle of that product. And I think that is where when I look at some of these findings from the inspectors, they don't see some of those evidences, and that's where they come up with the finding. So even though you may have followed some of the strictest principles of GCP, GMP, or GLP, if the inspector do not find them appropriately documented, unfortunately, it may result in the finding. And what does that mean? They may not trust the data coming out of the lab. They may not trust the data coming out of the clinical trial, where documentation was inappropriate. So that means they may invalidate the data, and you may end up running another clinical trial. For good manufacturing practices, it may mean whatever product has come out from that manufacturing premises, it may not be used in certain countries. And I think that's huge loss in terms of financial liability, as well as huge loss in terms of the reputation. And I think I can't stress enough the importance of documentation. So there is also element of data integrity. And if you look at the documentation, as I said, the documentation really should tell what happened, why it happened, if it happened correctly, what is that you followed. If it did not happen correctly, how did you course correct that, and what kind of root cause analysis principles you followed, what did you learn, and how did you follow that in the next part. So I think in any product life cycle, if you look at, you need to have documentation, so that even after 5 years or 10 years, somebody wants to look at that specific documentation, they should be able to recreate whole event as it happened at that time. And again, if you look at this is just an illustration of how that documentation means in that product life cycle. So, when you have that new medicine developed in the lab, you have that clinical developmental plan, then, you submit the dossier to the regulatory agencies. And during that clinical lab cycle, you also, document what are the adverse event or the safety effects are coming in, gets documented into that investigator brochure. If you have to design specific protocol, how that protocol should be designed, how the patient's rights are protected. So you design informed consent form. Once the study is over, how you are going to create the report, clinical study report. And this is all part of the documentation. When you conduct all phase 1, phase 2, phase 3 clinical trial, you need to submit that documentation for approval to the regulatory agencies. So, how you are, maintaining the documentation for submission to get that marketing authorization. And the story doesn't end there. After marketing approval, you also need to constantly evaluate long-term safety of that product, and make sure, either you take appropriate course correction in terms of future labeling of the product, in terms of putting any additional indication or additional information on the label, simple, like, maybe Orocore long-term uses. We have seen this drug is not maybe like safe for the patient suffering from cardiovascular diseases. So, for example, if this drug is to be given for a patient with oncology, if they have additional concurrent illness as diabetes or cardiac risk, maybe one should be careful in taking the drug, or do not take drug with empty stomach. If you can just hold on, because I have somebody coming at the airport and I'm just helping him. So I'm just going to put on mute for just for a minute. I just need to take the phone call.
No problem, sir.
Yes. Sorry about that. Actually, I'm helping one of my friend who is relocating with his family from Singapore. So he's at the airport. I sent the
Absolutely
taxi.
fine, sir. No issues.
So coming back, so if you look at after, so you also need to look at, for example, if you have to take certain drugs after meal or before meal. So some of this information may be available after the drug is launched, and you may have to adjust your labeling conditions. So this is all like the importance of documentation. And again, as I see, if you look at many companies sort of fail to provide that adequate documentation. And again, if you look at, we talked about some of the importance of the documentation. Again, the temperature control within the manufacturing premises, the good import. They also nowadays talk about good import practices. Are you importing information? So something which I realized when I started traveling overseas. In my first trip to the US, I was actually taking this wheat or jawar flour for one of my relative in the US. And I was checked at the immigration authority in the US, and I had to discard that material. Now, again, I was a bit surprised, because it is almost like food supplement, but what has to do? But I think there are some requirements laid down, especially when you bring any plant or animal product outside of the country of origin. Sometime, it may have some organism, even though there are some dead organism microbes, it will create trouble for that another country. So they also have specific requirement on what they call as good import practices, what you can bring in versus what you should not. Again, interesting example, when I was buying this house about 5 years ago, uh, the solicitor, the lawyer, actually, he gave me the history, that there was set up another house near the locality, and that has history of there is particular weed, a grass weed, which is called a Japanese knotweed. And again, initially, I was thinking, maybe why he's giving me that information. But the way it, then later on, I found that Japanese knotweed, it is type of a grass, and it grows particularly in Japan. Somebody took that seeds back in UK about 200 years ago, thinking that it grows very fast. It grows very fast, and therefore it is helpful for animals. But then I think that Japanese knotweed is so hazardous. When that grass starts growing, it grows in almost like 2-3 inch every day, and especially in the summer, it has ability to crack, pierce through the building walls, and that is quite, it can destroy your whole building. And that's where the UK government has almost like banned that, and you cannot bring some of these materials inside the UK. Maybe like how it is controlled in Japan, that is different. But again, this is just another example. There are certain requirements on what you can import versus what you cannot. So I think this is where now I'm just coming back what we studied in terms of that pharmaceutical product life cycle. We had, I think, we looked at where that life cycle journey starts, where it ends. Then we also started looking at quality systems, the Gxp, and then we mapped out those quality systems right from the birth of the drug all the way to its approval. And those systems we started talking about. So just to recap, we looked at what does the GLP or good laboratory practices means. We also looked at the GCP, the good clinical practices. We also looked at the good manufacturing practices. We also looked at the good distribution or good documentation practices, and the good import practices. And as we move along, I think there are tougher regulations. We talked about this US FDA ICH E6 revision 2 for good clinical practices, ICH E6 8, 9, 10 for GLP, GMP. There are also additional requirements, and most of these GXP quality standards, they have started utilizing technology, and using that technology to track changes in the processes, document their SOPs. And there is also another regulations you may or may not have heard of, 21 CFR Part 11 compliance. And that is something, as you use the technology, how you ensure all your electronic changes are tracked. So the concept of electronic signature or the digital signature. So how you ensure that those are the true signature of the individual who is documenting the information. And so they have put down some of those requirements. So the computer system validation. You also talk about in terms of how when you edit any document in the system, how you make sure that previous audit trails still exist. So there are some of this new sort of technology being used, and they follow this what is called as digital quality management system. And again, these are some new terms which are. I'm just looking at the watch. Hopefully, I should be able to finish in about maybe I think five to 10 minutes, and hopefully we'll have some time for Q&A. So if you look at in the quality system, they always talk about what we call as a RACI document, and it is a response. So for any process, who is responsible? And there may be more than one individual responsible, but who is ultimately accountable? And usually for any process within that product life cycle, it is one individual who is finally ultimately accountable for that process. For any process, who you can consult or who you need to inform. So again, and that is get documented. So just an example, if I talk about the laboratory practice. So for example, who is responsible for calibration of equipment? And that responsibility may be with 2-3 individuals, but who is finally accountable to ensure all the systems in my lab are accountable? Maybe it is the head of the lab. They may consult someone, maybe another lab or the accreditation agency to get that accreditation. So there will be some more than one individuals to be consulted, and they may have to inform somebody that my systems are calibrated. So they can be for example their customers. So for any sort of system in the pharmaceutical product life cycle, you will always hear this term called as RESI. Where is your RESI document? And RESI means they use this acronym, who is responsible, who is accountable for that process, who needs to be consulted, who needs to be informed. And then you have some of this quality by design principles. So again, how you inbuilt quality right at the beginning in the process so that your output is of good quality and you do not have to find something through the quality control process. So I think the industry call as the predictive quality management or proactive quality management, they call as quality by design. So there is an organization sort of, which is CTTI, Quality Consortium. So they are driving some of these principles these days across pharmaceutical industry. And as I said, quality by design, it is not meant for one Gxp standard. It is used in GCP, it is used in GMP, it is used in good laboratory, good documentation practices as well. And I think probably this is maybe like, this is my second last slide. So these are, I think, some of the approaches which are again defined. There is another guideline called as technical and regulatory consideration for pharmaceutical product lifecycle management, ICH Q12, wherein they talk more about the people, how that knowledge is stored in the system. So, if you learn something out of it, where it gets documented and how that knowledge is passed on to the next human being who is going to follow that process. And they use the term called as the established condition for manufacturing and control. In short, they call it as EC. So again, for the lack of time, I'm not going through that. But this is sort of my final slide. The way, if you want to sum up quality as such, it is not sort of a destination. The way as I see quality is a journey, and that journey never stops. And what does that mean? You constantly challenge yourself. If you have managed quality at certain level, how I can raise that bar next time. When I raise that bar next time, how I can go even further. And I think that is where I follow, I just differ in my own ways. Uh, each time you also need to ask that question, what is that I can improve? And therefore, there is some, you end up identifying some problem in any process. So what is going on? So you identify that problem. And once you identify that problem, you meet with that available data to perform analysis associated with that problem. So, and that problem could be anything related to manufacturing. Maybe, as I said, one of the problem is, yes, I have effective vaccine, but how that vaccine can be controlled better. So you meet with available data and then see how that can be controlled better. You conduct analysis, you identify various solutions, and then based on that, you own the best solution. You see if that solution works by verifying implementation. And then, when you complete that circle, then you think, what else I can improve better. So it is not that you're stopping there. And I think I just call that sort of, in my own ways, 8 watt methodology. So you keep on asking question, what I can do better each time through that product life cycle. And I think with that, Dr. Bhatia, that's what I wanted to say. So I know I spoke a lot, but I'm going to take a pause and see if any one of you have any questions which I can help answer. Dr. Bhatia, can you hear me?
Yes, sir.
Okay. So are there any questions which can help address? So I think that is all I wanted to say. So I'm just taking the floor here.
Thank you, sir, for your valuable talk and informative session. The session is now open for discussion. I request participants to unmute your mic and ask questions directly, or you can place your questions in the chat box or in the WhatsApp group also.
Participants, you can ask your questions.
I think so. It tells me two things. Either my talk was super clear to everyone, so nobody has any questions. No one understood what I was talking about.
No. Uh, I don't think there is either of the issue.
Right. Yeah. Yeah.
Sir, we have received one question here.
Please go ahead.
Can you tell more about ICH EC R2 guidelines?
Excellent question. I think let me share my screen again. Thank you. So I'm just going to share my screen once again. And probably. So, actually I have almost like 60-hour module which I prepared to talk about ICH E6 revision 2 guideline, and I'm going to finish that in 45 minutes for you. But ICH E6 revision 2, this is sort of an international guideline which needs to be followed by any pharmaceutical, biotech or clinical research organization while conducting clinical research activity. And these guidelines talks about how you can protect patient safety, how you can ensure data integrity. Now, just to give you a bit of background of those ICGCP guidelines, so when clinical research was conducted back probably 100 years ago, each and then again, typically this clinical research is conducted has been conducted in primarily in 4 regions: the United States, Europe, UK, and Japan. And each one of them, they had their own guidelines in conducting clinical research. So you can say a quality standards. And what they said that time, any pharmaceutical or biotechnology company who wants to register their product for marketing approval in their country, they had to follow this guideline, which was nice. So every company, they were following those guidelines, and they were getting approval for those drugs. So that's how some of these drugs what we are getting right now, right from penicillin all the way back paracetamol, or many some of this treatment like anti-cancer treatment, they have gone through that. But what I think everyone realized that was delaying the clinical research process, because and what does that mean? If I had to register product in the United States, US FDA has their own guidelines, what is called as 21 CFR, the Code of Federal Regulations, which is that determines what requirements should be followed by the pharmaceutical company in clinical research activity. So this pharmaceutical company will conduct clinical trial with protocol. They will submit the drug, the dossier. But then now let's say if the same pharma company wants to register their product in UK, then UK, they have their own regulations. So you almost need to conduct another set of clinical trial, generate the data, submit to UK, and then get the approval for marketing that drug in the UK. Then if they want to, if they want to conduct market the drug in Europe, you almost like conduct another set of clinical trial. And I think that was the case almost like 100 years ago. And very soon, I think thereafter, many pharmaceutical companies, even regulators, realized that is delaying the process. So instead of that, why don't, as a region, we come together where clinical research activities are conducted? So all these, regulators from the United States, UK, European Union, and Japan, they came together. I think that was back somewhere in 1990. And then they decided, let's come up with some consolidated guideline, consolidated standard. And then we give this standard to the pharmaceutical companies and tell them if any company follows this consolidated standards for conducting clinical research activity, and if they demonstrate through their documentation, the same documentation can be used to approve the drug in anywhere in the world, in across those 4 regions, and that they called that region as ICH region. And those guidelines then later on called as ICH GCP guideline, in International Conference on Harmonization Good Clinical Practice. So in short term, they call as ICH GCP. And those guidelines are defined in section 6. That's how it is called as ICH GCP E6 efficacy guideline. And in these guidelines, they talk about how you can conduct clinical research, how it needs to be monitored. These guidelines were first introduced in the year 1996. And that was they called as ICH E6 Revision 1. And that revision at the in year 2018, because of these technological changes, they had to look at these guidelines and make some changes, amendments. And that amendment came in force, which is called as ICH E6 Revision 2. So what is that ICH E6 Revision 2? It is built on some of the good clinical practice foundations. So it talks about some of the pharmacological aspects. It talks about the principles of GCP. So in some of the core principles, how the patient safety, data integrity should be protected. It also talks about the role of independent ethics committees in overseeing clinical trial activities. It also talks about role of regulatory authorities in terms of how they will be approving the conduct of clinical study as well as approval for marketing. It also talks about role of the doctor conducting clinical trial, role of the pharmaceutical company, and then there are specific elements, how the protocol should be designed. And I think I just talked about in investigational product, what are the considerations should be given, how that clinical trial should be monitored on site to make sure the data gets generated is of good quality. And then it talks about I think primarily that subject safety, data integrity, to make sure ultimately the rights and well-beings of clinical trial participants are protected and no harm is done to the patient. So that's is part of that ICH E6 Revision 2 guideline. Now, these guidelines, as I said, were revised in the year 2018. There is Revision 3, which is upcoming. That was to be planned, I think, in the year 2021 in the first half. But because of, I think, this COVID pandemic and other development, it got a bit delayed. But there is a plan to bring that revision in the year 2022 and part B of that revision in the year 2023. So what is that different to what we are doing now? So this ICH E6 Revision 2 guideline, talk about, of course, patient safety, data integrity, documentation. But as we are bringing this new technology, it is helping us to speed the clinical research activity. So if you think about, last year, clinical research program takes close to 10 to 15 years, and not clinical research, but whole product life cycle, right from the time you have something in the lab all the way to marketing approval. So that 10 to 15 years is too long. But last year, I think by using this technology, at the same time not compromising safety, we were able to get this vaccine development just in a matter of few months. So I think a lot of technology has been used, the concept of artificial intelligence, machine learning, lean principles you heard, and that has helped to speed the clinical research. But it also got some new challenges, like how you ensure the GDPR principles, the patient data protection. So there are, I think you may have heard about this Facebook or Twitter. They were fined by US government for leaking the data. You must be hearing a lot of political debate these days, how Facebook or Twitter, the data is processed. So in the same way, this clinical trial data, I think there are a lot of questions coming up. Just like if the patient is diabetic or patient is suffering from cancer, he or she may not want to divulge that information to somebody else, and just at the same time. So I think this GDPR, global data personal rights, or they call as an HIPAA principles, which is the PII or personal health information data principles regulated by the HIPAA regulations in the US. And also, I think just last week I heard China is coming up with personal data protection bill, which is going to be introduced on 1st of November. So that is becoming more now important, even though it has nothing to do with clinical trial. But clinical trial involves exchange of personal data or patient data. So how that data is protected so that that still remains confidential and we are not divulging any unnecessary information. So those factors are getting considered, and you will see that gets incorporated into that revision 3 guideline. So I know it was a long answer to the short question, but hopefully that gives you the flavor about ICH E6 revision 2.
Thank you, sir. So we have another question here.
Yeah.
Uh, at academic level, is it possible to get into clinical research for newly developed products?
Uh, Definitely, yes. And I think, if you look at, and I'll give you an example. Uh, if you think about this COVID vaccine, and recent example is AstraZeneca. This vaccine was developed at Oxford University. So if you look at most of this drug development, actually, it is part of maybe academic research or part of industry academia association. So it is absolutely like some of these activities. So Oxford developed that vaccine, and then they needed sort of infrastructure. That's where the AstraZeneca came in. So they provided their infrastructure, and then you remember, like they also needed, because one of the challenge of this vaccine development last year, we needed this vaccine for 7 billion population across the globe at the same time. And there is no manufacturing facility in the world who can produce that vaccine in a shorter period of time. The largest facility is in India. That's why they ended up tying up with Serum Institute as sort of giving the technology transfer, make sure vaccine gets developed. And you must be hearing a lot about some countries getting vaccine, another is not getting vaccine, but it is not because I think one is raw material availability, and even though you have raw material available, there is no such facility who can produce those vaccines. So always, I think this is where it is one of the new positive learning from this COVID pandemic. The academia like this Oxford University, industry like AstraZeneca, and the big manufacturing facility like Serum, they came together. And even there was a very interesting article. While this was happening, they still found that this production is not adequate enough. So even though sometime you have some of the pharmaceutical competitors who would compete for marketing sort of similar medicine, they came together. So likes of like Sanofi, they gave their vaccine facility in Europe for production of this. Merck, they also gave their vaccine facility to produce this vaccine. So I think there is a lot of encouragement. And and again, coming back to your question, there are a lot of universities engaged in these activities. So even if you look at this company BioNTech in Germany, it was started by two scientists, husband, wife, and you will be surprised that they still live in a small apartment. And then they developed this vaccine, but they didn't have enough infrastructure. That's how they tied up with Pfizer. And now they have got billions of dollars, and of course it is not a profit, but and now they are repurposing that money to develop vaccine for HIV. And this is the, I think for HIV, I think we are trying almost like since 1980, the pharmaceutical industry developed vaccine. It is not, but they have got the ray of hope. So there are different technology, mRNA, and I think some of these technologies sort of, students like, some of the students you have, and maybe some of the guides like you do that research, and maybe out of 100, one idea clicks, and then you need that additional support. So I think that is where, if you remember the slide that I showed you, the initial part is not regulated, but when you find some promising molecule, this is where then you have seen many industries, many universities tie up with pharmaceutical companies. Even I'll give another example. Have you heard of this called as Operation Warp Speed? So this was another area last year when COVID pandemic hit. Everyone realized you do not have enough time. And so At the same time, many people realize that we need huge funding. So that time, again, I'm not talking about Donald Trump or others because it is highly political, but I think the US government and pharmaceutical companies, they came together on the 20th of March 2019, and they said like, now this whole world is grappling with COVID pandemic. We must need to do something. They also invited universities, and they decided what is our ultimate goal. We need to come up with 300 million doses of vaccine by 1st January. You have 8 months in hand. Now you tell us what you need. And then that time, you have some of the scientists from the universities, some of the regulators. They also said, now you tell us how we can formulate the norms so that we make it easier for you, but at the same time, we are not going to cut the corners. And that's where, even there were some investors came and they generated close to $8 billion US funding. And that funding was given to these companies like, Merck, AstraZeneca to develop this vaccine. So and this is where some of these the academic research turn into sort of industry research. So if you look at some of these vaccines which are getting the origin started in some of this academia. So yes, I think to answer your question, it is possible. And again, these guidelines are not, in my opinion, I consider as a rocket science. It is sort of ultimately common sense. If, as I said, like why we do celebrate the World Pharmacist Day? Why do we celebrate Clinical Trial Day? I think it has some simple logic. So same way, if I have to come up with a good clinical product, what kind of quality system do I need to put in place? And the way I would like to think again, in vaccine development, there is a lot of talk going on, even including my mom back in India. She was telling me, don't take vaccine. And it's like, it is not safe. People are saying it is done with an act to reduce the population. People are likely to get impotent. And I think, this is something, the question I would like to ask. I was part of this clinical research program for some of these vaccines. I know how stringent guidelines has been taken. And I'm asking that question, can I take that vaccine myself? Can I give it to my dearest one? And if my answer is yes, that's the quality standard for me. So I think if you follow that, then that is exactly following the GCP guideline. That is exactly following GMP guideline. Even in my previous life, I was working for a the IV parental company, and they had their logo saying that, what is my quality standard? If I'm preparing one saline, I ask that question to pass that test. Can I give this to my daughter? Can I give this to my father in case they're ill? And if my answer is yes, my product is passed. So hopefully that gives you sort of another flavor in terms of those activity and how that has close link with academia.
Okay, thank you, sir.
Yeah, anyone having any questions?
Yes, ma'am. Sir, as far as quality systems are concerned,
yeah,
generally we came across the terminology Six Sigma inspection model.
Yeah.
So means it is only stick up to the 6 digit only, or it is much more than that?
Excellent question, Mr. Mali. I think I was hoping somebody will ask that. There was deliberate attempt done by me not to touch upon that topic. So we talked about GCP, GMP, we talked about GCP. I also talked about lean very silently. So this is the concept, lean and Six Sigma. Now, again, it is sort of what I realize in sometimes industry, they are fancy of using some of this very big jargons. That's lean or Six Sigma. But again, you understand what is Six Sigma. So it is something how you can reduce the defect in your system. And the way, you should not have more than 6 defect in 1 million operation. And again, 1 million operation means 10 lakh. So again, so if you are, what does that translate? If I'm going to produce 10 lakh tablets, I should not have more than 6 defective tablets. Now, what that defect means, my tablet should be of certain size. My tablet should look good. There should not be any black spots. My tablet should pass the QC test in terms of, having that active ingredient between 98 to 102%. So those are your quality standards. So what does this Six Sigma principle says? You should not have more than six defective tablets in the 10 lakh tablets that you're manufacturing in the lab. And that is maybe in a simple way, Six Sigma principle. Now, the Six Sigma, again, if you look at, you also hear the term lean and Six Sigma. What does that mean? You reduce the waste in the system and make your product quality of the same quality. So this concept actually was originated by Toyota in the car manufacturing industry. So, basically, and then it was later on, taken by Henry Ford. So typically this concept started in the automotive industry. And what does that mean? If I have to manufacture a car, and there are several parts in the car, like you have a gearbox, you have tire, you have sort of different controllers. So each of those parts, how I can make those parts in such a way that there should be less defective parts. So that's where they started that concept, and now that has become sort of an how you can apply those Six Sigma principles in other type of industries. So those concepts are very good, but one thing we need to remember: you cannot apply same concepts in the same fashion in any industry. So what I mean by that, you can look at those six defective operations, but then you need to operationalize that, the type of industry that you are working on. So that's sort of the Six Sigma concept. The Lean Sigma, it talks about how you reduce the waste. So what they talk about, if as a part of risk management, things are going to go wrong, how you can prevent something going wrong. And I'm just trying to give you a simple example, probably. And I do let me try to elaborate. So if probably it may be there in Kolhapur or in Mumbai, Pune, I have seen that. But when you go into a big multi-story car park, so usually when you try and enter the multi-story car park, there is sort of a U-shaped angle, like almost like your car passes through that gate, and that gate has certain height. And that height is just above the height of your car. Now, and again, sometimes that gate is, that bar is not sort of very thick. It has rubber cushion around that bar. Now, again, if you think of, there is a logic behind that. What they say, you want to avoid the damage to the building by big truck entering inside that gate. And even if that big truck enters, that we do not want the truck to be damaged. So that's why that rubber cushion. So this is something you're trying to avoid waste. You're trying to avoid defect by putting that simple thing. And this is something they call as reducing the waste, or sort of being innovative. Again, I'll give another example, but apologies. Hopefully, that will make more sense. And I laughed myself. In Amsterdam airport about 10 years back, I was traveling and I went into the urinal, and they had a picture of fly, a black picture of fly in the urinal at the point where you urinate. And I was just amazed, why is that? And then I came back and I started reading. I have that curiosity. I started reading what does that, because at that time I was reading this Lean and Six Sigma principle. And then they, basically they realized they wanted to keep all toilets neat and clean, because that is one of the busiest airport. So when you look at that black fly on a white portion, you try to aim, and therefore you do not pass urine at the other parts of the urine. So again, it is a simple concept. You try and reduce the waste. You have to clean that less times. So again, these are very simple concepts, but that has those principles originated from Lean Sigma, Six Sigma. Again, the Lean Sigma principle talks about another way of reducing waste. Can I do some operations in parallel, which is what was followed in vaccine. So if you remember again, in vaccine, when AstraZeneca and Oxford University, they found this promising molecule, they started developing that through that animal studies. They found it effective, but at the same time, they realized, whole world is, almost like one single delay in bringing that vaccine, it is costing 100,000 patient life. So they took a risk and then they started manufacturing vaccine while clinical trials were ongoing, thinking if the clinical trial passes, I do not have to wait for another 2 months to manufacture that vaccine. So there was a risk of few million US dollars they took, thinking if the vaccine was not going to be effective, whole batch or efforts would have gone west. But then they're thinking of doing processes in parallel to speed up the clinical research. So again, I think so that is where now it has given new sort of norm. So again, giving one more example of this Six Sigma or Lean Sigma principles. You think about this data management we used in the clinical trial, what is called as patient diaries. So again, typical example, if patient has to report adverse event or any side effect, they come to the hospital during normal, say their routine visit, say every week, and then they will tell, I had fever last week. But then doctor will document that fever into the case report form, which will be analyzed by a pharmaceutical company a month later. So the fever which was actually experienced by the patient 6 months ago gets analyzed almost a year later. And then in that whole clinical research program, you then talk about 100 protocols, and that's why it takes like 10-15 years. But in this case, what happened, we had electronic diaries with the patients on their mobile app. So as soon as patient said, I have taken the vaccine and I'm getting fever, they entered fever on the app. Immediately the data was connected to the hospital. Doctor immediately got alerted patient had fever. That fever, it was immediately analyzed by the pharmaceutical company. The pharmaceutical companies, they have their operations running 24/7. So even though it is middle of the night in the US, someone in India is working in analyzing that data. Even though it is middle of the night in the US, in India, someone in the US working on that. And then, so that's how the fever which was reported by the patient within same time it was analyzed. And then you can imagine maybe there are 30,000 patients who were reporting that data across the globe. Their data was analyzed in real time, which used to take almost like good 6-7 years to analyze that data. And then that's how that whole period shortened. These are the concept of like bringing system, making systems lean. So you got that, brought that technology, and all your additional steps cleaning that data got crunched from almost like 6 months to few seconds. And I think this is what I meant. These are good concepts, but then you need to think how you operationalize that into your own setup. And this is where I'm saying, how that ICH E63 guideline came in, wherein now, because we also have to think about how we protect that patient data. So how you ensure that patient himself is reporting the symptoms and not somebody else. Otherwise, that data is not valid. So this is where now everyone is started thinking about how you assure that data credibility, how you ensure that the same patient is entering the data, not somebody else, and how you manage that by not seeing by the patient. And that is the new world, what we call as a decentralized clinical trial. And this is one of the application of, by the way, excellent question, Lean and Six Sigma principle, bringing into pharmaceutical product development.
Thank you, sir.
Sir, we have received another question from participants.
Yeah.
To be acquainted to GMP and GLP of industry, what extra information need to be given to B.Pharm or M.Pharm students in colleges?
Yeah. And I think this is another excellent question. And with due respect to all my, teachers, well, I think most of you, this is something, in my humble opinion, what I have realized, there is a gap in what is taught in academia versus what is needed in the industry. And I say that because I spent close to, in the last 25 years, I have conducted probably more than 150 workshops, and I'm saying on site. And out of my own interest, I also started looking at what did I study in my MPharm curriculum 25 years ago versus how much of that I'm applying today. And to be honest, my answer is sadly different. Of course, that basic knowledge really helped me, but the the concept that I'm applying, you talk about Lean Sigma, you talk about Six Sigma, you talk about quality standards, the GMP systems. I don't think, some of this knowledge is taught in the academia. And I think that is the sort of gap. Now, that doesn't mean, basically, you need to be worried. I think whatever knowledge you have, that's still good. And something, again, this is the power of technology. Right now, I started, managing my new role, decentralized clinical trial. I have good I believe I have some knowledge on the operation, how clinical trials are done, but I lack that technical expertise. And I have started, like, if I just go on Google search and try and just say how this data analytics needs to be done, there are good 5-minute, 10-minute tutorials, videos, which can give you the same knowledge without you spending money, or at the same time help you to acquire the skills. So definitely, I think some of this principle, as I talked to you about, what is this ICH E6 guideline say? What is this ICH E10 or E9 guideline talks about? I think that is the information you need to acquire. In ideal world, if it gets incorporated into the curriculum, that's excellent. But I think there are some roadblocks, to be honest. I made that futile effort last year to design one of the MPharm in clinical research curriculum for a couple of universities in India, and even approached a couple of universities in the UK. Maybe because of the level of bureaucracy, and I think you have to go through several layers. It is a bit difficult, but slowly I think there is a realization, and then now I think people are realizing. And again, take an example, the current program that you are running, you are inviting some of this industry, so you are gaining that knowledge. So again, to give an a simple answer, if you look at some of this concept and start seeing, yes, if you there are some certification available if you want to acquire those skills, but not necessarily you need to go through that route. You can also learn on your own, and I think that's another. I thank COVID for we some of the positive experiences we learned from COVID. Like we have brought this technology, and we are able to connect the people regardless wherever you are.
Yeah, that's very true, sir.
Yeah.
Yeah, I'd like to ask a question from my side, sir.
Yes, please.
Just to know, I think during your clinical trial phase, are these efficacy and safety guidelines of the ICH taken into consideration?
Sorry. Yeah. Sorry. Can you please come again? I'm sorry. I was multitasking. Yeah. I'm sorry.
No, it's okay, sir. Just wanted to know, during the clinical trial studies, like for maybe like the COVID vaccine came into, or maybe for any other new formulation which is coming into the market. So when efficacy and safety guidelines we have to take into consideration. So how does it work with when the clinical trials are going on?
Yeah. So I think these guidelines, and I think this is something. If you look at the efficacy and safety guideline, ICH 6, how it works, there is also a new concept which is called as adaptive clinical trial design. So, initially one of the way, and again, these are some of the concepts of applying Lean Six Sigma principle. Initial way was, when molecule comes from the lab, you think based on the chemical structure, this molecule might work in, say, oncology. And because that's how, and then you try and work, and then after 3-4 years you realize probably it is not as effective as you thought, and therefore then you take U-turn and you say, okay, but probably during the same time, this is almost like a discovery. You realize maybe this molecule has promise in diabetes or hypertension. So then you come back, start from phase 1 and start doing some additional clinical trial in hypertension. You proceed further, and then you realize maybe like it is doing well, but in specific group. Then you come back, and then but realize maybe it has another implication. So you almost like review each indication one by one. And now this is new concept of adaptive clinical trial design. There you almost like run parallel studies in the same protocol at different dose levels. And this is where you try and understand the efficacy, safety, and you need to be extra careful. And actually, I have in my another computer, there was a fantastic case study in the year 2006. There was unfortunate tragedy. You also need to be extra careful. while doing such studies, what happened? The dose which was found safe in animal, that dose was increased, I think, by 2 folds. Oh, sorry, it was like 1/10 of that animal dose, and then it was phase 1 study. Couple of patients developed brain damage, and then there was a big press release. And I think this is where they tweaked those guidelines. Now you can basically you still need to do these controlled trials, and when you design the dose, make sure you have solid justification. So you cannot compare. Of course, we are from the monkey family, but monkeys still, their habitat is different than what we are. So is that 1/10 safe dose, or it should be 100 and 100th or 500? So I think that is part of that. Coming back to your question, Dr. Bhatia, efficacy and safety. And I think it is a bit interesting, because on one hand, just like I'm taking example of last year, we know several patients are dying. We do not have enough time. We need to come up with the vaccine, but at the same time, there is clear ask. You cannot cut the corners. So the drug needs to be safe. If the drug needs to be effective, at the time it needs to be safe. So interesting, you said, and many times people feel none of the vaccine which has been is in use right now, it has received full approval. They call it as emergency use approval, EUA, because still all these vaccines which are given, their post-marketing or data is collected, and it is part of that safety and efficacy analysis which is conducted. And maybe very soon some of these vaccines will get that final approval. And this is how you may have seen as a part of this ICH safety and efficacy guideline. About, I think, in the month of February, the AstraZeneca Janssen vaccines, it was put on hold. Why? Because suddenly they realized there were some thrombolytic events occurred, and there were about 16 deaths. Now I think when you hear about this, then suddenly some of these layman people say this is the reason we were telling you shouldn't take vaccine. But then again, as a part of that efficacy safety parameters, when you analyze that, they found these 16 deaths were amongst that 76 million patients, the people who took the vaccine. So almost close to 760 lakh individuals who took the vaccine, there were 16 deaths. Now the 16 deaths, when you analyze the data again as a part of that safety and efficacy analysis, they realized the 16 they were from age group below 30 years of age group, and then they also found they had other concurrent illnesses going on. Somebody was suffering from cancer, somebody was diabetes, somebody was hypertensive. So the death couldn't be attributed to the vaccine alone. And at the same time, they found there was another age group between the same age group, but without those illnesses, it was absolutely fine. So again, that's why what they did, what they decided, they came up within a week saying that we have done that safety and efficacy analysis. We know this is safe, but as an additional precaution, what they decided, do not give AstraZeneca vaccine below 30 years of age group. For that, again, in the UK, they started, for above 30 years of age group, they started giving Pfizer, Moderna vaccine. Below 30, above 30 years, it was AstraZeneca or anything else. So again, I think, if you go back, and I did a bit of research, and if I look at the first vaccine which was launched in the global market, polio, and if you look at the potential adverse effect, side effect, number of deaths occurred, there are more than 16. So you also need to look at, I think, one of the parameters in safety and efficacy analysis, they look at risk-benefit ratio. So when you look at risk-benefit ratio, and I think this is more important for an oncology study, and we had a lot of debate. This is the role of ethics committee. Somebody asked the question. So if the patient with cancer, which is terminally ill, and unfortunately, it is the question of when that patient is going to die, 3 months, 6 months, a year. I know it is very sensitive, but in that setup, if there is a ray of hope, there is a molecule which has potential to increase the life expectancy. Then you look at the same time, molecule safety is not established. So you see the risk of giving that drug, which potentially may help increasing the lifespan. Or so that is the benefit, and risk is it will reduce the lifespan. Instead of 3 months, it could be less than 3 months. So I think this is sort of in a risk and benefit ratio which is calculated in the clinical trial. And then in many cases, when the benefit weighs the risk, that's where the clinical trials are started. So again, we have seen in some cases, most of this drug, when they're given, it helped in improving the lifespan. Of course, some of those patients unfortunately died, but instead of dying, say in 6 month time, their disease progression prolonged. So they were able to live with their loved one at least for a year, in some cases 2 years. And that's part of the journey now. So again, I think it is a long answer, Dr. Bhatia, to your question. But it's like part of that, doing that safety, the efficacy analysis, and considering risk and benefit ratio.
Sir, even when the drug repurposing is done, say suppose one of the drug was being tested for one of these, like you said, in case of cancer, and later on you just come up to the case that probably it is more useful for diabetes or hypertension. So in this case, whenever suppose you stop your trials with respect to the cancer treatment, and suppose now you want to begin it with for diabetes. So, do we have to repeat the concept of safety and efficacy, or you can just procure the data from the earlier study which was conducted for cancer?
It's again, thanks, Dr. Vata. It's another excellent question. And I think this is also another way how, as a pharmacist, we can give assurance to the public, like, when the drug comes, it goes through a series of vigorous testing. Of course, still sometimes things fail, but again, this is an experiment we learn from. So to answer a question, there is a requirement. Again, it is part of the ethical guideline requirement. So if you look at this 21 CFR regulations, ICGCP guideline, any pharmaceutical company, they have to provide the results of every single clinical trial, regardless it is positive result or it is negative result. And when I say positive results, yes, we had initial hypothesis which said, this drug, we think this drug will work in oncology. We tested, we designed the protocol, we analyzed the data, and now we have found it has promise. So we want to additionally conduct some more trials. So it is a positive outcome. But let's say if it is negative outcome and it resulted in not improving that health. So they still need to publish that, because that is just in the spirit to make sure drug failed, it is not efficacious, and also it is still safe. And if you have established the safety, then you may want to try it outside. Let's say in your example, or the example that I gave would be diabetes. But in that case, diabetes, things are different. Now you're not treating oncology patients, you're treating diabetes patients who have sort of a different sort of pharmacological parameter. Maybe like they're on different type of medication. You do not know that medication and this study drug, how that interaction is going to happen. So you still need to add and ask some of those questions, and then look at safety from a different point of view. The safety is established in an oncology setup, but not necessarily in this kind of setup. So within that phase 1, they call as phase 1B. So again, you may not need to go through animal studies because that initial safety has been established. So you still need to look at that and make sure if the drug was found unsafe because of whatever, say like it is causing, and just an example, increase in blood pressure or brain damage. I'm just making that up. Then that means probably you should be careful even trying that out in any other indications. So I think again, to answer your question, first part, you need to see why the drug failed in the previous indication, whether that drug failed because it was not the right indication and drug is still safe. It was not effective there, and therefore now you want to try it out. And when you try it out, think about these drug-drug interactions. Look at that. And interestingly, again, you may have heard last year there was a lot of black population in the US. They refused to take vaccine, and they made a lot of sort of I think the sort of road shows saying that this is the, and there is a reason. If you look at back, I think it was somewhere there was a Tuskegee syphilis study conducted about 800, I think 400, 500 years ago. And unfortunately, this was an academia study. One of the academician, unfortunately, he ended up, because in the clinical trial setup, it is an experiment and you don't want to risk. So they ended up enrolling all the black population. And then that syphilis study, they developed a lot of congenital deformities. And then later on, it was found that it was purposeful. So they just chose particular population. So therefore, they refuse. So basically, you also need to look at in that clinical trial setup, are we engaging that homogeneous population? So if the drug was sort of safe in white population, is it safe in, and therefore you may end up again just looking at that phase 1 trial from a different perspective. There's a lot of talk going on diversity and inclusion in clinical trial. Actually, I gave another talk and I looked at the data which in the year 2000, from the year 2015 to 2020, 5 years, whatever data was submitted to the regulatory agencies. If you look at that data, and I'm talking about data not for one clinical trial, across all indications. You talk about variety of indications. There were close to 200,000 patients participated in different type of clinical trial in that 5-year period. Surprisingly, 80% of patients were from white population. Only 11% were Asian. And if you think about that, those drugs, when get launched into the market, it is consumed by, if you just put China, India, and Indonesia put together, that these are the world's populous country amongst those top 5. They have close to 45 to 50% of population. So the drug which is consumed by 50% population, only 11% of the patients were studied in the clinical trial. And that's where I think there is a lot of talk going on. You need to have this clinical trial more inclusive, more representative of ethnic origin. And therefore, some of these new things are coming up, even in the phase 1 trial. Are you including homogeneous population?
Thank you, sir. Avni, you can go ahead now.
Thank you, ma'am. It was really nice and informative session, sir. Now I request Mrs. Sujata Chaudhary, ma'am, to sum up the session.
Is it audible, Shivani?
Yes, ma'am, it is audible.
Good morning, one and all present here. It's my privilege to propose vote of thanks on behalf of all present learners for this AICT-sponsored faculty development program, Pharmaceutical Quality System for Product Life Cycle Management. The session was very interesting and informative. Sir has explained all the concepts very nicely, like how pharmaceutical product journey takes place, what are qualities of pharmaceutical products and how it can be raised. Sir has explained concepts like quality by design, quality risk management with various examples nicely. Sir has explained how protocol design in clinical research takes place, how problems can be identified and how it can be overcome by available data. Sir has explained what are various quality standards like good manufacturing practices, good laboratory practices, good clinical practices, and good documentation process along with its importance. Also, good distribution practices, various parameters that involves. Sir has also summarized various ICH guidelines and its importance, and also emphasize on importance of clinical trials too. The current scenario where quality system for maintaining product life cycle management concept is really very important, and thus this session will definitely helpful to all present learners. Lastly, I want to thank you Principal Bharati Vidyapeeth College of Pharmacy Kolhapur, Maharashtra, Dr. H.N. More sir, Vice Principal Dr. M.N. Bhatia sir, Co-Coordinator and Head Dr. N.M. Bhatia ma'am, Co-Coordinator Dr. D.P. Mani sir for organizing this faculty development program. Definitely sir, it will helpful to all of us. Thank you once again sir. Thank you all of you. Thank you sir.
Thank you.
Thank you, ma'am.
With that, Dr. Bhatia, I'll take your leave and
Yeah.
wish you all
Thank
the
you for
best.
your
Yeah.
yeah.
Wish
Thank
you
you
all
so
the
much
best
for
for
your
your
time, sir.
program, and thank you.
Yeah, thank you so much, sir.
Madam, just a minute.
Yeah.
Participants, I request you to turn on your camera so that we can have a photo with our eminent speaker.
Sure.
Is it done, sir?
Yes.
Thank you so much, Patil, sir.
Thank you. Wish you all the best, and hopefully we'll see you in person in one day when I'm back in India. Thank you.
Yeah, definitely. Hope to meet you soon, sir.
Thank you.
Thank you, everyone. We will begin with our second session shortly.
Who is she? What happened? Why you are sleeping? अरे बापरे. आपल्याला नाही का ते? येताना एवढं गडबडीत यायचं घरून येते ना रूमवर असते पाहून यायचं चालतं इथं बसायला तरी आहे